So what I'd like to, I guess, talk about today is a mix of old and new. The first part is just pick one topic from some old stuff that touches on some issues that I think are still interesting, and that attached that lead into, in some ways, the main body of the talk, I think, which will be new. And both of these topics have to do with how does the fact that evolution occurs in a spatial context affect the fates of deleterious mutations. So let's just start by that context that almost every species exists in space. I don't mean space like George Clooney, but space like spread across the landscape. And the individuals with them are spread over space. And these can be fairly complicated. It certainly can be a lot more complicated than we like to believe in our models. This is they can be spread over islands. The habitat can be complicated. This is some eco map of Wisconsin. This is just in here to prove I used to do field work. These are fork fungus beetles that live on patches. Populations can go extinct, recolonized. This was originally put here as just an excuse to show my formerly cute kid. I guess he's cute now, but he's not this size. He's this size. But to remind you, that he went to daycare. And how many of you guys have kids that have ever been in daycare? Right. So as a biological experiment that they go there and they come back with all the germs, right? And you get all the germs. So there's this daycares are basically source populations for human disease microorganisms, and we're the sink populations when they bring them home to us. There's this adirectionality of flow of organisms from one direction to the other that causes a kind of complex spatial and temporal structure of that. So there's a lot of, and this just hardly even touches the kind of complexities that can happen. Populations in space can be quite complicated. One of the ways I like to think about populations in space is to think about not just differences between places, but similarities of individuals at the same place. So individuals that are near each other tend to be more similar to each other than they are to randomly chosen members of the rest of the species. And that similarity extends to a lot of different fairly obvious things, but these things interact in ways that become interesting. They tend to share similar habitats, right? So they might be evolving to adapt to local conditions, perhaps. They also share similar genes. So individuals that are nearby are likely to be related to each other, at least in the diffuse sense that we think of relatedness at a population level. They're more likely to be genetically similar to individuals that are nearby. And that means because to the extent that competition for resources is local, you're competing with relatives for those resources. To the extent that mating is local, you're mating to a certain extent with individuals which are related to you. And in general, any kind of interaction that's spatially limited is tending to be interactions between relatives to some degree or another. And so there's this feedback between the genetic similarity and the environmental and social similarity that creates a lot of context by which evolution can be different in a spatially explicit population compared to the usual things that we might see in chapter 1 of a population genetics textbook that leaves that out. Okay, so that's by way of introduction. I want to try to address two fairly specific questions today. So one is, how is selection against deleterious mutations, I should be in here, different in spatially structured populations? I have no idea how Madonna does it with this thing, because it's completely, she must get used to it, right? Do I? Right, should I do that? That wasn't very good, was it? Sorry. All right. Oh, this is on freaking YouTube, isn't it? Oh, geez. Okay. Next time you see me, next time I start to go off the page, just somebody signaling. Okay, and this is the new part of the talk. How has selection changed in the spatial context of an expanding population at the leading margin of the population that's expanding into a new territory? How does that that specific moment of population expansion over space affect the way deleterious mutations might be patterned over space? Okay, so let's start by thinking about selection in these metapopulations against deleterious mutations. So this is part of a larger project that dealt not just with deleterious mutations. And in general, I was trying to look at some of the simple population genetic quantities and that we perhaps knew a lot about in a panmictic single population, and start to think how that might be different by space. What I was trying to do was use quantities that were measurable in nature to varying degrees of success. I wanted to allow for the kinds of demography that went beyond just the sort of island model that's typically the starting place for these kinds of analyses, and kind of with that reduce some of the hidden assumptions, like the assumptions about what kind of population structure. And for this general realm, it turned out that with these three parameters, you can go a pretty long way in understanding some of the effects of space on selection. One was, the first and the third are the ones that are most important to us today. One's the partitioning of genetic variants, so how differentiated are different local populations. I'll define that more precisely for you in a second. We're going to measure that in terms of a quantity called FST. Effective population size, how much genetic drift there is. That one's not so important in terms of what I'm talking about today. And then finally, what's the relationship between local genetic fitness and the local productivity of the population, right? So in other words, who are you competing with for access to resources and to be able to put your offspring in the next generation? I'll tell you more about what I mean by that in just a second as well. Okay, so this FST. So FST is a very commonly used measure of genetic differentiation among local populations. There's a million different interpretations of FST. One of them that's particularly useful for what we'll talk about today, this was originally defined by Sewell Wright back in the 40s. And this is a quantity that looks at so this Q here is the allele frequency of a local population. So you have for each population, you have a local allele frequency, and this is the variance among that list of allele frequencies over populations. P bar and Q bar are two alleles, and their average frequencies across all those populations. So this is a standardized measure of the genetic variance among populations, this FST. Okay. FST is also, it turns out, a measure of how much extra homozygosity you expect to see in a population as a result of the spatial population structure. So FST also measures the proportional excess of homozygotes caused by the non-random mating associated with spatial structure. Okay? So both of these interpretations turn out to matter in what I'm going to show you in a second. Okay, so FST is a measure of how different populations are. It also tells us something about how similar genes are in the same population, which is what leads to this homozygosity. Okay. All right. Now that last thing about the relationship between productivity and fitness. The usual axis on which people talk about this is in terms of hard selection and soft selection. You know, hard selection and soft selection are some of those words that even if you hear them, almost nobody remembers what they were, and even if they do, what you were taught they were might be different from what somebody else was taught they were. So we'll just start. I'm going to tell you what I think they mean, or what I mean for the next 10 minutes by them. So hard selection is when the productivity of a population is proportional to its fitness. Is this the one thing you wanted a picture of? Okay, good. So if you have a higher relative fitness than somebody else, it doesn't matter who's around you, you're going to have a higher fitness. You're going to leave more offspring in the next generation. Okay? Soft selection, the productivity of a population is independent of the genetic fitness, relative fitness of the individuals that are there. In other words, with pure soft selection, you're imagining that there's perfect local competition for resources, perhaps, so that a population creates a collection of individuals has the same number of offspring collectively in the next generation, regardless of how good or bad its genes are. Right? And so you might imagine that coming about because, like I said, there's local competition for resources. If everybody's really good, they partition those resources out. If everybody's really bad, they partition those resources out and make the same number of babies. Okay? Now, this still allows for selection among, if there's a good individual and a bad individual in the same place, the good one will have more offspring than the bad one. So there's still a possibility for selection, but it's all local. Okay, there's a lot of complexity here. I could say a lot more about this, but won't. But I'd be love to talk about it if somebody cares about this stuff. Okay, so here's just a cartoon of that. So with, and I put them in the wrong spatial order, sorry. But so soft selection on the top here, you've got two populations. That's the clusters over here. And this is an extreme case where there's complete genetic identity within each population. These guys are all red, these guys are all white. Selection, the red guys are better. They make 50% more babies than the white guys. But with population regulation, there's only 6 spaces, so those guys all go back to make 6 red guys. These make 6 white guys, and then that's the whole story, right? There's no change due to selection in the allele frequencies in that population. Now, had I started with some variation here, there would have been change, but it would have been the selection within populations that engendered that change, right? So I'm taking an extreme case. With hard selection, start from the same condition, make the same number of babies. Population regulation is global, or there's no population regulation, if you prefer to think of it that way. So there's 50%, what did I say, 50%? Twice as many babies. So you make twice as many offspring relative to the other population. So selection has been effective there. It's not been constrained by the local competition, right? So soft selection is this inherently unfair but natural thing. Lots of things can be unfair and still be real. This inherently unfair thing where if you are good, you're likely to be around other good genotypes, and they eat up all the food, and that means there's not that much left for your babies so if there's this positive correlation in genotypes across space, and there's local competition for resources, that's going to limit the amount of response to selection that can be possible in a population. Okay. All right, so the broader project addressed lots and lots of different issues: mutation load, inbreeding depression, fixation, partitioning, blah, blah, blah. I'm really going to focus just on the first 2 of these things here, the things that relate to deleterious mutations. Okay, so I'm going to, this approach allows for fairly complicated population demography, but we're going to zoom in on a fairly simple genetic basis for fitness. And that's a single locus. We're going to look, with just 2 alleles. One allele is good, one allele is different, better, worse, depending on whether S is positive or negative. And the heterozygote is somewhere in between, and where they are in between is defined by the dominance coefficient H. Okay? The math for this assumes that selection's weak, that the strength of selection for or against these alleles is weaker than the amount of migration between populations, and that just makes some of the approximations come out a lot easier. So that's assumed as we go. Okay, now before I started this, I'm thinking, there's actually 2 things that could happen here, right? So there's 2 kinds of things that I could think of that would affect the way selection worked in a structured population. Even with this simple, by the way, the selection's so simple, those are the fitness coefficients everywhere, right? There's no heterogeneity in the environment in terms of how selection works in this model, right? This is spatially uniform selection. Okay, so the response to selection, how much does the population change because of selection? In a structured population, it could be decreased by this local regulation of population density, i.e. soft selection that I was just talking about. So soft selection makes selection less effective, so it would decrease the response to selection. But there's another thing that does the opposite, and that is when you have a structured population, you've got for the same allele frequencies, you'll have variation among populations in those allele frequencies, and that has as a consequence that the probability of a locus being expressed as a homozygote is greater in a structured population, and it's greater in proportion to FST. Okay. Now the thing is, homozygotes are more different from each other than either one is from the heterozygote. And so by expressing things as homozygotes, you're actually increasing the genetic variability that selection can see. Okay? So that makes selection more effective. Okay? So, could go either way. How does it work? That's what I want to try to get at. Yeah, picture. Okay, so Sorry, I shouldn't do this here at NIMBioS, but I'm just in the habit of doing it. So math. Here's the answer. Sorry, that's really not what you wanted. So this is the change in allele frequency under pure hard selection. This is the change in allele frequency under pure soft selection. These are approximations that basically the only thing we need to assume is, like I said before, that selection is weaker than migration. It includes the sort of PQS that you would see in a panmictic population, but it also includes effects of FST and dominance, which would have been there in a panmictic population as well. So with hard selection, you have this extra elements in the equation that come from FST, and I'll show you the consequence of that in a second. Basically, if you stared at this equation long enough, you'd see that with FST, that gives you a stronger response to selection because you're expressing more homozygotes. Okay, so with hard selection, there's no soft selection counteracting. You get the greater expression of homozygotes, more effective selection, typically. With soft selection, you've got all the same thing that's happening above. So just there, look here, delta Q hard, just the same thing copied down there. But on top of that, you have a term here that captures the effects of soft selection, the competition between relatives. So basically, this term amounts to 1 minus the relatedness of individuals that are in the same place. Right? So selection is less effective because of competition between relatives that occurs with soft selection. Okay, so let's look at this graphically. So I'll show 2 or 3 graphs that have the same structure. FST will be on this axis. So FST goes from 0 when there's complete absence of any effect of space on the gene frequencies. There's no spatial pattern. And gets higher as you go to there's more and more variation among populations as you go to the right. Probably in here is the zone that's most important on this graph if you're thinking about population structure within species. Okay, over on the left-hand side. Okay, so in each of these graphs, I'll have FST on this axis. I'll show you the relative value of something over here. In this case, of the frequency of the deleterious allele at mutation selection balance. So this is the equilibrium frequency of a bad allele, divided by what it would have been in a population that had FST of 0, divided by what it would have been in an unsubdivided population. Okay, so that these are all going to converge at 1 when FST goes to 0. Otherwise, I screwed it up, but I'm pretty sure I didn't. All right, so that's, okay. And then I'm going to show you cases with different values of the dominance coefficient. So H equals 0.01. The deleterious allele is nearly recessive. The heterozygote has only a 1% of the effect of the homozygote to nearly codominant here on the top. And there's solid lines that show us what happens with soft selection and dashed lines that show us what happens with hard selection. Okay, so with hard selection only, we get more effective selection in a structured population. There's more homozygotes. Selection can see these deleterious alleles better and remove them more effectively. That's particularly important when the alleles are recessive. They get highly expressed. So deleterious alleles are usually rare in panmictic population or any population. Deleterious alleles are usually rare. They don't come together and form homozygotes that much in a panmictic population. So selection, if that's the only place selection happens, it's not very powerful. In a structured population, even those rare alleles are expressed as homozygotes relatively often. Selection can see them, act against them, remove them from the population. So the frequency becomes much, much lower with either hard or soft selection for nearly recessive alleles. Okay? With nearly codominant alleles, hard selection makes them go away better. Soft selection is less effective because of this competition between relatives. So it depends on the details, but there's a lot of cool insights if you stare at this long enough. Okay. Have you stared long enough? Should I go? Yeah, okay. So, okay, now let me focus on something called mutation load. So I just showed you the frequency of deleterious alleles. The deleterious alleles that are present in a population cause a reduction in the mean fitness of that population. Okay? And that by the amount by which mean fitness is decreased by the presence of those deleterious mutations, we call mutation load. Okay, load's a concept that will come up again and again through the talk. Load is a reduction in fitness caused by some feature compared to the case where that feature doesn't exist. Okay, reduction in mean fitness. So here, it's due to the fact that there are deleterious mutations. So reduction in mean fitness caused by these deleterious mutations. Okay, so now this gets a little more complicated again. We tend to have a lower frequency of deleterious alleles, as you saw on the previous slide, in a structured population. But they're also expressed as homozygotes. Okay, so the actual fitness consequences for the individuals that carry them may be a bit larger on average in a structured population. So how does that interdigitate? Well, that's shown here on this slide. How much, Cordola. It's not shown on this slide. There's a slide missing. It looks kind of like this. There's a reduction in mutation load that goes down to about 50% in the case of the recessive alleles. This slide is the next one. Sorry. This one tells us how much inbreeding depression do we expect to see in populations as a consequence of the population structure. Okay, so inbreeding depression is you take individuals, mate them with relatives, ask what their fitness of their offspring is relative to individuals that are outbred. Okay, when you do that calculation, you get a fairly dramatic difference. When you have population structure, you can get for alleles that are nearly recessive, you can get dramatic reductions in the amount of inbreeding depression caused by those loci. Okay? That's mainly because those bad alleles that cause the inbreeding depression are rarer in the population. Okay. Now, you don't get such a dramatic effect for codominant, but let me show you, hopefully, yeah, I do have this slide. Okay. When we think about inbreeding depression, the amount of inbreeding depression contributed by a locus is largely a function of dominance coefficient. Okay? So for the smaller the dominance coefficient is, the more inbreeding depression there is. That's because if the dominance coefficient is a half, the heterozygote's just halfway between the two homozygotes, it doesn't actually matter to the average whether you express them as those same alleles as homozygotes or a heterozygote. You get the same average fitness. But where the alleles are recessive, and where the deleterious alleles are recessive, when you make more alleles homozygous by inbreeding, you're creating exactly the combination that's bad, that's creating inbreeding depression. So it's when they're recessive that you get the most, it's when deleterious alleles are recessive that you get the most inbreeding depression. It's when deleterious alleles are recessive that you get the biggest effect of population structure. Right? So structured populations, among other things, are probably very important in determining how much inbreeding depression there is in a population. Okay. All right, so we'll just take a kind of brief summary before I go on to the next bit. In subdivided populations, you'll have to believe me, I didn't show you, but mutation load tends to be reduced in structured populations, and inbreeding depression is likely to be much reduced in structured populations. So the spatial context and the nature of competition between individuals matters a lot for what we expect to see for these deleterious mutations. Yeah. Mm-hmm. This is equilibrium.
And so if you initially started off with the population, starting inbreeding, you'd actually expect
yes.
you'd be paying that very large cost of the load.
That's right. That's right. Now, if you had a panmictic population and then you structured it, then you got inbreeding depression that's reflective of what the allele frequencies were because it was panmictic. Yeah. Yeah. So this is not if you were a wildlife manager, you would not say, "Oh, I'd rather have a population that's fragmented. Let me do that," because that would be a disaster. That would take the problem of the old and inflict it even more strongly. Yeah, that's right. Okay. Right. Okay, so let's, here's the new part. I want to talk again about an explicitly spatial model. This is about populations expanding over space. The classical example of this is actually European humans spreading out of Africa across Europe or across Asia. I guess that would make them not European humans, but humans spreading across Europe or Asia. That can create interesting and sometimes counterintuitively strong patterns in gene frequencies over that space. Now that's something that's been investigated before. The particular thing that we bring new to this is we want to look at the interaction of the dynamics at the front of the wave of expanse with the dynamics of populations that are adapting to a heterogeneous environment as they spread. Okay, so let me show you that in 2 bits in a minute. I'm going to start with what people call the acknowledgement slide, but really this is the author slide. We decided to do this project. Basically, our whole lab joined in to do this project, which I highly recommend if you have a lab or if you ever do in the future. I know we were at NIMBioS and nobody has labs, but if you did, just getting a whole bunch of people together, even if it's totally inefficient, it's a lot of fun and everybody learns a lot from each other and you get a lot of benefits beyond just the writing the paper thing. And so we've done it. Most of these people are in my lab. Amy and Anna over there on the right Amy's a professor at UBC and Anna's a kind of free-roaming postdoc at UBC. Jeremy's a former postdoc. Fred is at University of Zurich and was previously in my lab a long time ago. But so we just a lot of people, way more than we needed. But it was just stopping here to say this is really a fun way to do things sometimes. Kim's name is in giant letters here because Kim's done most of the actual computation work on the project. So if we scale these things if we scale the pictures proportional to contribution, it would look different from this. But so this is more than half Kim's work. Okay, so how do deleterious alleles accumulate at range edges, and how is this affected by heterogeneous environments? That's my main question. Okay, and I'll tell you why we're asking this here. I guess starting now. So this is a figure not from our work. This is from Stefan Peschel, who's now at University of Bern, working with Laurent Escoffier. And Laurent especially has been instrumental at raising our awareness of a phenomenon called allele surfing. And I'll show some cartoons of how this works in just a second. It's that alleles can reach high frequency by genetic drift at the leading edge of a range expansion and the consequences that come from that. This is a paper where Stefan and Laurent looked at the fitness effects of that, where deleterious mutations might fix by chance, right, against selection, but drift overwhelm selection. So just focus on this right side here. This is space going across here. Population's expanding over time, which is this axis. The population is expanding over time. And then there's a spatial pattern at any point in time where there's a lower fitness at the range margin. And in this case, that's nothing to do with the environment. The environment's uniform everywhere. This is because of deleterious mutations fixing at the wave front. And I'll show you more about why that happens in just a second. Okay? And you can get fairly dramatic fitness differences they've shown. Now, this is a lovely paper. There's 2 or 3 papers that they've done. They make a lot of kind of abiological assumptions in their parameters. So one of the goals that we have in this is to kind of crank up the biological realism of the parameter sets. Right? So that's kind of our sub-goal. But our main goal is there's completely uniform space here. Okay. Now, there's another line of population genetic thinking about range edges that date a lot back to this paper by Kirkpatrick and Barton in 1997. There's been a lot of work since then, but this is just a figure from there. And the notion there is that if you have spatially heterogeneous selection, in particular, if your optimum, this axis on each graph is space, if this is the optimum, the solid line here is the optimum phenotype with a quantitative genetic model, and this is the density of individuals at any particular point in space. And what they found in that paper was that if that optimum varied over space strongly enough, the population would not spread. It would collapse to a single point or go extinct. But if that optimum was weak enough, if the change of that optimum over space was had a slope that was low enough, the population could expand indefinitely over space, right? And basically in this model, or sorry, under this parameter set with the weaker slope of the environmental gradient, populations would just continually adapt at their range edge. They would always be doing more or less what they were supposed to do at any point in space. They would have enough offspring to expand and let the population continue to expand. In this case, they fail to respond. Everybody's got the same mean phenotype, essentially the same mean phenotype everywhere. And as a result, you get to a point where your fitness is just not high enough for the population to replace itself, and it can't sustain a population beyond a certain spatial range. Okay? So in this body of literature, we have a changing environment that may constrain range expansion or the rate of range expansion. Okay? So these are two really kind of cool aspects of population genetics, both about range expansion. So basically our research project started from, well, what if we took these two things and collided them, right? What if we put these together? What would it look like? And I'll tell you more why we care about that other than just let's see what happens. We had some reasons for wanting to do it. Okay, before I do, I just want to, expansion load might be something new to some of you, so I just want to go through some cartoons to show you probably where this stuff comes from. Okay, so I'm going to show you just a series of cartoons. This is location. You start from a population that's over here on the left, and it's spreading to the right. Okay, so that leaves this kind of classical wave of expansion pattern of density of how many individuals that are present in any given slice of the population. Okay, now these expanding populations have edges, and they have unusual features. So the edges have two features that distinguish them from the rest of the population that make them particularly important, and these two facts collide to create expansion load. Okay, so one of those things is that the edge probably has a low density. Okay, so there's just individuals that recently got there. It hasn't had time to expand to full density. If it had, the range edge would be further along. Okay, so there's low density. That means there's a high potential for genetic drift locally in those populations. Okay, on the other hand, the other fact about the range edge is that if you have spatially limited dispersal, which you do if you're talking about species edges, if you have spatially limited dispersal, those guys on the edge are best placed to be the source of colonists for the empty territory out in front of the population, right? So the most likely ancestors of the future population that'll be over here are the individuals that are here on the edge, somewhere here on the edge. Okay, they have two features. One, drift, because they're small population size, and two, they're super lucky in that they're close to all this empty resource that they might expand into in the future. Okay, so those collide. Imagine that a mutation appears, somewhere, perhaps near that edge. That mutation may drift to a relatively high frequency, and if it does, it may actually locally fix. If it does, as that population spreads, that allele is the allele that gets carried with the expanding population, and you wind up with this regional fixation of a new allele, of a different allele. Okay, so just parenthetically, one of the reasons that this allele surfing is important is this creates a pattern that looks just like what local adaptation looks like genetically, right? So if you said, oh, look, this variant is really common in the north and this variant's really common in the south, that must be local adaptation. No, there's a huge way that populations can look like that from a completely neutral process. Okay, but from our point of view today, these alleles that spread by drift may not be selectively neutral. They may actually have consequences on the fitness of the individuals they carry. In fact, they might be deleterious. So deleterious alleles can, in fact, fix, if they're weakly deleterious, in the population through this allele surfing. Okay, and that's what I want to focus on today. Okay? Okay, now, so as I said, I wanted to collide these two things, this allele surfing, expansion load. Oh, by the way, expansion load is the load caused by the expansion, right? So it's the reduction in fitness caused by this regional fixation through allele surfing. Okay. Yeah, so I want to see, how does this expansion load collide with this sort of Kirkpatrick and Barton world where there's local, where the optimum varies over space? Okay. Right, now if you think about that for a minute, again, it could go either way. Okay? They could positively interact. It could be the case that you've got poor adaptation at the edge to the heterogeneous environment. That may reduce the affected population size, cause you more drift, cause you more expansion load. Right? That a priori seems totally plausible to me. Or it could be that there's a negative interaction, that the poor adaptation at the edge may actually slow down how fast the population expands. And if it did, that gives more time for good alleles from deeper into the range to catch up, right? So that it minimizes the spatial catchment from which the ancestors of the future range expansion may come, because there's more mixing as you go. If you go slower for the same amount of dispersal, you get more mixing. Okay, so which one is it? It's the second one. I'll show you in a second. Okay, so this is a hard problem. We don't know how to do math for this, so we did simulations. This is all simulation project. We used a program called NIMBio that Fred Guillaume developed. It works pretty well for this. We have a two-dimensional landscape. It's 40 units by 2,000 units for our population. I'll tell you, units don't matter. You can call them kilometers if you want. We have a core here that's 40 by 40 that we let sit there for a really long time to burn in and reach some reasonable allele frequencies before we start the expansion. Okay. The populations then expand from, I'll just show you left to right on all the slides. They expand from left to right, and we stop when we start getting individuals that reach the far side. So we're not wanting to have any edge effects. We're just assuming that we stopped it somewhere before it kept going forever. Okay. There are in total about 500,000 individuals that would fit onto this landscape if it were fully at carrying capacity. Within a sort of typical amount of dispersal distance of the edge, there's enough room for a couple thousand individuals. Okay, if you took the slice within a sort of typical dispersal distance of the edge, there's room for about 1,000 individuals in that range. So we're not talking about super small population sizes here, even potentially at the margin, although the actual number of individuals that are at the edge is lower than that because of the patterns of density over a range expansion. Okay, so dispersal. So mainly, so this kind of technical detail. NIMBio is a program that assumes that everybody lives in a particular population. And so we've kind of tricked NIMBio into doing continuous space by creating a grid of places, using that grid for a discrete approximation of the continuous dispersal function. So if we wanted to have something like a Gaussian dispersal curve, we have a very finely, we have a dispersal curve that's actually got 16 elements in it to make something that looks a lot like a Gaussian, but it's by bins. Okay? So at any given square, any given population, there's a carrying capacity of 6, but they can disperse over approximately 200 cells. For a typical individual could disperse to roughly 200 cells. So the discreteness of this doesn't, I think, matter so much. It's a way to trick. It's hard to do continuous population genetics on a simulation. We've tricked it into doing it by this discrete approximation. Okay. Our baseline, I think actually everything I'll show you today is based on a Gaussian dispersal kernel. We also did this with kurtotic dispersal with long tails. Right? So they had a reasonable probability of going twice as far with a mixed model of two Gaussians. They don't actually differ in any of the main things I'll show you. The last slide, which shows how fast they spread across the landscape, the kurtotic dispersal makes them go faster. That's the only place it'll appear because they didn't look different. Okay. All right. Now, what do we do for selection? So we have a quantitative trait. Quantitative trait has Gaussian selection, right? Stabilizing selection around the optimum. There's a strength of that selection around that. We have 100 loci, each locus that affect the quantitative trait. Each one has the possibility of mutations. We track it with a real number, the effect of that locus, of that allele. The effects of any given allele mutate according to a Gaussian addition of an effect. Okay, and with a given parameter for that mutation. So we've tried to match this to the best estimates we could find of how much mutational input is there for a quantitative trait from empirically estimated values. And that's true for all the parameters on the selection functions. We assume, just like Kirkpatrick and Barton, that there's a linear gradient over space. Okay, so everywhere sort of up and down has the same optimum, but going from left to right, you're changing the optimum. Either you're not changing it with this b equals 0, b is the slope of it, or you're changing it. This b equals 0.0375 is about enough that you get about a 0.5% loss in fitness by going a typical dispersal distance. The 0.375, you get about a 5% loss in fitness by going one dispersal distance, right? So fairly weak, kind of fairly moderately strong change in the optimum. Okay, and then deleterious mutations, we track the genomic mutation rate either at 0, so just leaving out deleterious mutations. This is completely separate from the quantitative trait. They're on the same genome, but different loci. Either we don't do it, 10%, which is kind of what you might see in Drosophila, 0.1 deleterious mutations per genome per generation, or 1, which is the order of magnitude of us humans. We're probably more in the order of 2 or 3 deleterious mutations per genome per generation. It's kind of horrifying. We have 1,000 biallelic loci. For each of those, we drew a selection coefficient from a gamma distribution that is this distribution. This, again, is parameterized from empirical work. We have a dominance coefficient that declines with S, again, motivated by empirical work. So the weak effect mutations are codominant, and they go to nearly completely recessive as the selection effect gets stronger. Again, that's what people see in nature. Okay, so all this we've been trying to, all right, so let me just show you an example run. Here's a population that's starting to expand. Each of the lines here is tracking the frequency of some deleterious mutation. The darker ones are stronger effect mutations. And I'm just going to click through going across over time. You can see that there are deleterious mutations that fix locally. And of course, once they fix locally, the only way for them to be repaired, well, we do allow for back mutation, but the easiest way for them to fix is for the good mutation from over here on the left side to catch up and spread through the population. So if you actually track some of these as we go through time, you'll see any given line tends to move a bit to the right as well because selection is pushing that wave across. But we get a fair number of deleterious mutations that wind up fixing in these populations. Right? These dark blue ones have a selection coefficient of at least 1%. Okay. We thought it looked like this famous picture. Now, I'm just going to highlight this one that I've now colored. It was there before, but I colored it in orange. This guy turns out to actually have a selection coefficient of 9%. Now, this one's cherry-picked to show you, this is one we noticed, like, oh, wow, that's cool. It's not a typical run. But there's an allele with a 9% effect, and it's regionally fixed. And so here, when you get nearly to the end of the spread, half of the population is fixed for this really horrible 9% deleterious allele. That's really strong. And as you go over time, the spatial pattern of that changes. It spreads as you go. Okay, so let me summarize. I know I'm supposed to stop soon. But I should probably tell you the results before I do. So we do get a reduction in fitness due to these deleterious mutations at the range edge. Okay. So these graphs show what the core population is like compared to what the edge is like. So the edge, in each case, when there are deleterious mutations, it's worse off at the edge than it is at the core. Okay. I'll show you something about the line in a second. I'm going to reparameterize this in terms of the expansion load. How much reduction in fitness is there in the edge relative to the core? That's plotted here. Well, there's none when there's no mutations, right? That's just doesn't need to be on there. But you can see there's nearly a 50% reduction in fitness by these parameters at the edge relative to the core caused by this expansion load. So the kinds of things that Pyshal et al. Are seeing are entirely reproducible with, I think, somewhat more realistic biological parameters. Okay. Right, so it's about 50% reduction. Now, this was just kind of a sidelight. This wasn't why we were doing it, but it was nice to recreate what they did in this sense. It's also the case that you have local adaptation reduced at the range edge. Again, we expected to see this from previous work, but in the edge versus the core, you can get substantial reductions in fitness caused by the fact that you don't have the right quantitative trait at the edge. It hasn't yet adapted there. But when we add expansion load, right, so when we add these deleterious mutations in, that reduction of fitness caused by maladaptation is not nearly as bad. Right? So by having the expansion load causes the load due to the quantitative trait to be not as great as it was going to be. So in this case, the fitness is roughly 50% greater than it would be without those deleterious mutations. The fitness from the quantitative trait is about 50% greater than it would have been without the expansion load. I'll tell you why in a second. We can look at I think, where are we at? Oh, right. Let me go back to this slide here. The same is true in reverse. So these different cases are how strong we have the gradient for the quantitative trait, how much, and the fitness with the gradient, with the strong gradient, from the point of view of the deleterious mutations, is a lot greater than it would have been in the absence of that gradient. And in fact, there is a massive change in the amount of expansion load, massive reduction in the amount of expansion load when you're adapting on this spatially heterogeneous gradient to when you're not. Okay, so this is the same thing's happening in both directions. If the population's dealing with load from one of these aspects, the load due to the other aspect is not as bad. Okay, and here's why we think that is. Both of these things, both expansion load and environmental heterogeneity, slow the rate of population expansion. Okay. Complicated slide, but it does, right? If you got lower fitness at the edge, the population is not expanding as fast. Okay? And that slowing expansion allows two things. It allows the beneficial alleles from behind the margin, from the population as a whole, to reach the edge and ameliorate some of that expansion load. If you slow the expansion because you've got a maladaptation of the quantitative trait, you've got more time for the good alleles to catch up for the deleterious loci. If you've got expansion load caused by the deleterious loci, you've got more time for that population to adapt to local conditions on the edge. Okay? So what we think is that the slowing of the expansion creates a situation where evolution can be more effective to ameliorate the differences between the edge and the core. Okay. And if anybody's curious, here's just a quick slide to say that for that expansion load, what kinds of genes cause it? It's actually genes that are of intermediate effect, which is something that we expected because of general results on drift load, but it's quite dramatic, right? So there's mutations here in the 1% to 2% range that under these parameters are disproportionately responsible for this expansion load. Okay, so in a real population, these are things we might actually, if we're lucky and have a large enough study, be able to measure and detect that they exist. Right? And for that matter, they can have, like in a human context, these deleterious mutations that may have come about by expansion load may have large enough effects to notice in the individual health, the health of an individual, and maybe even strong enough that you could do something about medically. And remember, the thing is, these are locally fixed. So this is not like regular mutation load where everybody's got a different set of deleterious mutations. Everybody in these descendant populations have the same deleterious mutations, making it a more statistically tractable and medically tractable thing. Okay, so this is the last slide. We can get a substantial expansion load even with realistic parameters. Stefan and them have had greatly increased the realism of their parameters, but this is a different set. The adaptation to spatially heterogeneous environments and expansion load interact. In particular, if you have local maladaptation, it reduces the amount of expansion load. If you have expansion load, it increases the ability for the population to locally adapt at the edge. And we think both of these are because of the slowing. Each of those sources of load slows the progress of the range expansion and allows more time for evolution to do the right thing. And with that, thank you again, and thanks Sandy again for bringing me here. And if you have time for questions, I do. Thanks.
Okay, do we have any questions? We have to use the mic.
So it seems one way to look at what you're talking about is there's a kind of a time scale quality to this, right? Like what is the migration rate from the core per generation relative to sort of the expansion rate? And so if you had a fluctuating, instead of this gradient in the environment, if that was fluctuating up and down, right, and it slowed the expansion, then you would see the reduction of these effects. But if that fluctuation sped up the expansion, then these reductions of loads wouldn't be seen. I guess I'm confirming my intuition here, but I guess the real question is, it just seems like it's very much a time scale question.
Absolutely, yeah. No, if you wait long enough under these conditions, everywhere will locally adapt.
Okay.
And all those deleterious mutations will eventually get pushed off the right edge of the thing.
So why should we care? Here's my question.
Right.
So who cares?
Well, you come from European descent. You, on this same time scale, have ancestors who spread from Africa into Europe and potentially bringing in these same deleterious alleles, these kinds of deleterious alleles. If you have if you care about conservation biology and you plunk down a population in a new area and it expands, there could easily be a reduction in fitness in that expanding population.
If you waited long enough, eventually you would expect that. So
Yeah. If long enough is possible. Yeah. Yeah.
Mike, back to kind of human examples.
Yeah.
Is it right that the longer you expand, for example, like looking at Native Americans, right? So we've been expanding the longest period of time, and Southern America was colonized relatively recently, right? 10, 15, whatever, 5,000 years. So what are the implications? I mean, like if you start comparing deleterious mutations like in African populations with those in Europe where expansion was relatively short to those for Native Americans. I mean, are there any implications?
Yeah, there might be. We haven't looked at it in that way. I can't tell you what the. I can kind of tell you. We have looked at it, and I can't remember I don't have the slide to show you. But I can't really remember the answer. But the mean fitness doesn't go like that. I think it tends to. There tends to be a drop off in mean fitness at the edge. Don't listen to me. Sorry, there are possibly ramifications. I can't remember what the answer is. So we have looked at these things over a spatial pattern of mean fitness in exactly the way, as a function of the distance from and I just can't right now remember what the answer is. So I just shut up. So I will. Sorry.
I had a question. So what if you had a species that had, I guess, greater capacity for phenotypic plasticity, or I guess some other mechanism in which they could handle steeper environmental gradient slopes? How would that change any of the patterns that you've seen from your simulations?
I'm sure it would. I'm not sure how. Yeah. So I think as far as I know, the faster the things expand, the more of these problems will happen. All else being equal, right? One would imagine that if it buffered by phenotypic plasticity and therefore allowed to spread, it would potentially be accumulating more deleterious mutations as it went. Yeah. So I guess the only direct thing we have about that, so the original model by Stefan and them had soft selection I should say this is all hard selection, well, hardish selection. At the edge, it's hard selection that, in the sense that you're not automatically granted the right to have the population spread. It depends on the ecological fitness depends on the genetic fitness in this case. And that's much less conducive to expansion load, right? If you just spread without regard to your genetic qualities, you get way more expansion load and it extends much further back into the population. So I would use that to say that it's likely something similar might happen if you had a buffering from selection caused by plasticity or the other things you mentioned, that would likely cause the problems to be more pronounced.
Kind of have a related question to what I was asking. So one thing is the distance, but also like the slope is very important, right? In your model. So like south-north dispersal or just kind of east-west. I mean, the gradient presumably will be much stronger if you go south or north. So what would be the consequences? Again, like if there is a population that is spreading, kind of staying in more or less the same.
Oh, I see. You mean if the selective gradient was perpendicular to the expansion? Yeah, I don't know that would have. I don't know what effect that would have. This is all where the gradient goes in the same direction as the wave of expansion. That's all we've done. If you had a gradient that went this way and an expansion that went that way, I imagine the effects would be much more subtle, but I don't know. It'd be interesting to look at, I think, because there may be less mixing, and therefore you might have more effective local drift. The locality of the drift may be spatially constrained along the wave of expansion and not just relative to the core. So it may have a different effect, but I think it'd be a less strong effect than what we're seeing here. That's just conjecture. Thanks, Sue.
So have you considered putting mating system in here?
Nope,
And
I have not considered. Well,
would you? I mean, you can
yeah.
imagine that you would have very different dynamics depending on.
Yeah, so I think something that would be really interesting, and I spoke to you, and we have actually talked about doing this, is looking at pollen seed dispersal. And the reason I think what would happen in that, under what I, as a non-botanist, think of as the usual case of pollen dispersal being greater than seed dispersal. Did I get that right? Is that good? Fair enough. It depends. Yeah, okay. Anyway. But in that particular case, where pollen dispersal was more widespread than seed dispersal, what would happen is that the seed dispersal would determine the rate of population expansion, but pollen dispersal would determine the rate of mixing from the core to the edge. And I think you get a much, much lower amount of expansion load in that case. But we haven't looked at that explicitly.
Right, but I was talking more about like mating system differences, like mixed mating or self.
That'd be interesting. Yeah. I mean, so the thing about selfers is they don't have. Yeah, that would be interesting. I don't know what would happen. We haven't looked at that. Yeah.
So going back to the human example, you
Yeah.
can have humans that just kind of spread out via diffusion, but you can also have really different methods of movement. Like if you look at the American West, where they kind of stopped the Mississippi River and then jumped all the way to California and then went back and kind of filled in the rest of it. So how would you handle something that didn't have really a clear wave, that had something that was more kind of leapfrogging?
So by they,
around?
you mean Europeans coming into North America?
Yes.
Yeah. So I think you'd have to model that explicitly. I mean, this is obviously a very what we've done is a very reduced cartoon representation of the kinds of things that might happen. So with particular human populations, we sometimes know enough about the history or prehistory or other things to have a more demographically explicit model. And I think the Escoffier group has done some of that. So yeah. Uh-huh. Oh.
He also mentioned his study with sample operators of colonization of Canada, like Montreal area.
Mm-hmm.
He was saying something about very negative results, and he couldn't even find any evidence of his recollection.
Yeah, there's it gets really complicated, and I don't know all the I haven't made up my mind who's right about this, but you get very different answers from the human geneticist about whether there's genomic evidence of frequencies of deleterious mutations, and it's that varies. Some people say you do, some people say you don't, and they're working from the same data, so it's I just don't know that's not the Quebec thing that you're talking about there, but just even spreading through Europe per se. There's lots of different answers about what you do and don't see.